And well, this webinar will be about basically my work, but not just like my studies in general, but the topic of how we can use genomics to actually answer questions in conservation and biodiversity. So this will be just like a framework where we can actually use all of this new information that we have from genomics to really answer real life questions in conservation biology. So let's get things started. And the main thing is when we look at, today, genomics is perhaps the biggest field in biology. It's really has gotten off, and it's a revolution in its own mind, and it has had this great impact on both medicine and agriculture. And like no one is doubting that. And the reason why it's really had this great effect on medicine and agriculture is these are perhaps the two topics that we as people are really interested in. Medicine is important for our health, and genomics is now a big part of that. And breeding or agriculture is a part of both feeding, the ever-increasing populace. So really, these two applied fields of biology have really had a great impact from genomics, and we can do a lot of things, and it's got a lot of emphasis. And with that, genomics has also, there's like really great analysis pipelines. There's cutting edge research being done in these areas, and genomics has really supplied a lot of information to these two topics. But there's actually another field of biology, an applied field that is very important for us today, and that is conservation biology. Because as we know, because of global warming or other reasons, environment is changing. And as environment changes, as ecosystems change, we are dependent on these ecosystems. We're actually dependent on a functional ecosystem to actually, continue living in this world. So to protect that functional ecosystem is also very important, and that's where conservation biology comes in. But when you look at conservation biology studies, although genomics has a lot to offer this area, it's only been used in a very limited kinds of ways. So it's not, I don't mean that genomics hasn't really been used in conservation biology, but it's been very limited when you contrast it to all the developments that have come out in medical and agricultural research. For example, here's one study that we did in the last few years, which actually shows how typical biologists really use conservation biology. That is to obtain a lot of genetic information and to look at genetic diversity on a map, or look at genetic diversity in a geographic context. And this is fine because this can really tell you a lot of information. For example, the question here was how many different units do we have, and especially this black species, whether this black species here designated its, or this population here designated its own conservation unit. So when you look at the overall genetic structure, you can actually see that the black population is really much more diverse than all of the others of this fish population. So yeah, perhaps we should actually differentially protect or build another kind of conservation based on it having high genetic diversity. This is, an acceptable way of using genomic information. But sometimes, and most usually, biology is not that simple. So here's another example, which we did. Again, this is about conservation of fish, of longfin smelt along the Pacific America coast. And when you look at these, the question here was, were the populations in the Bay Area, the ones in red here, were under danger? Because as you know, San Francisco is a big city and there's lots of pollution going on. And so the question was, of all of these different populations, was this Bay Area different unit that we needed to conserve? What was the diversity? Was it under decline? What was the population rate and whatnot? And all those kinds of questions. And again, when you look at genomics to inform something about diversity, you can actually see that the Bay Area population is really close to most of the other populations, except for the ones high up in Canada. So basically, this just means that we don't really have a separate population in the Bay Area, and it can also always be fed by these other populations. So it's not really, even if populations are declining, there's not really much problem when you look at genetic diversity. But this is just a very simple way of looking at it, and it doesn't really give us most of the information, because when you use genomics to actually understand the evolutionary dynamics of the species, it gives you a much different picture. For example, we did a modeling using approximately 200,000 loci to actually observe migration rates and historical migration rates between populations. And what this actually showed us that all throughout the past, the Bay Area was the single population that always fed the other populations via migration. So basically, the Bay Area population is the source population, and all of the diversity of all of the other populations actually comes through this Bay Area population. So even though genetic diversity in this Bay Area population is high, and even though population numbers is high, this doesn't mean we don't need to conserve it. We really need to conserve it because this is the one population that actually feeds and is a source populations for others. So this is just one very nice example, very simple example of how we can use genomic information to actually go beyond simple patterns of genetic diversity and actually infer evolutionary dynamics. And that can actually tell us something about the conservation policies that we should actually enact. And of course, genomics has a lot to say about evolutionary dynamics. So when we look at evolutionary dynamics, we've known for a very long time that if we have genetic information, if we have information about a gene, then we can use this to actually track evolutionary history. So we can use genes to measure divergence time between populations. We can use it to measure changes in population size through time, and a lot of different dynamics that occurred in the past. But another thing that we learned very quickly is that every gene usually has a different story to tell. So if you have one gene, when you build a phylogenetic tree, or when you do an evolutionary, like a molecular clock, when you actually do an evolutionary history using these genes, that is the history of the gene. So you're actually modeling gene divergence. And gene divergence, we quickly learned, can be different from population divergence. So there's a difference between how genes diverge and how populations diverge. So if you have one single gene, and if you're trying to estimate population processes from this gene, you might not always be correct because the gene might not really be a good representation of the population history. What you actually have to do is you have to sample a lot of genes, and then through these genes actually try to estimate the most accurate population history. So what we want to do ideally is have information from a lot of genes to use that to infer the population history as best we can. Because what we want to model is not gene divergence, but population divergence. Only when we have a really good idea of how this population divergence is, only when we can really model population divergence as accurately as possible, then these estimates of like divergence rates or effective population sizes or speciation models are actually accurate. When we do these on specific gene trees, they might not be so accurate. Now, in the past, this was very difficult for evolutionary biologists because, sequencing one gene, two gene was, pretty hard work. But this all changed, about in the early 200, like around 2010, with the second generation sequencing revolution. So what this, supplied, as we all know, is it made gene sequencing very easily. We no longer had to sequence each gene by itself or each individual by itself. We could just do this in massive parallel sequencing. So we could pull a bunch of individuals together, and we could sequence all genomic information from those individuals at very fast rates. And actually, technology is getting much cheaper. So today we can cheaply sequence genomes from hundreds of individuals in one single run. So basically what the second generation sequencing allows us as evolutionary biologists, it gives us information from all of, from the genome. So no longer do we have to work with one or two genes, we can actually very easily work with all of the genomic information. And this really helps when we're trying to model evolutionary history to answer specific questions. Of course, being an evolutionary biologist is not really easy, and especially when you're trying to do conservation biology, because there's still a heavy cost involved. So when you're working with model organisms like, Drosophila or mice or all that kind of stuff, you usually have a lot of money. So a lot of these fundings go to these model organisms. So it's specifically easy to sequence genomes from a lot of individuals. But when you're working with this, with conservation biology or with natural populations, you usually don't have that much funding. But of course, the best part of next generation sequencing is that it's pretty flexible. So whatever your budget is, you can find some sort of way of getting genomic information. For example, if you're doing conservation biology, priority should be to have a lot of populations and a lot of individuals. So a good strategy would be to actually increase the number of individuals, increase the number of populations, but for example, instead of sequencing whole genomes, get genomic sampling. So not sequence the whole genome, but just sample the genome from certain genes. For example, get information from 20,000 loci or 200,000 loci or something like that. And this is actually genome sampling for natural science. For example, when you're working with natural populations, is a really good way to balance your budget. It enables you to get genomic information. It also enables you to keep the budget at friendly costs. But of course, with this said, most probably this problem of funding will be moot in about 10 years, when we will most probably have, sequencing will get much cheaper, and so it won't be really a problem to sequence a lot of individuals from a lot of populations. But today, most people working with natural populations usually work with some sort of genome sampling, and that's what I wanted to have a shout out here for it. And these are some of the first examples which use genomic data for evolutionary inferences. For example, when you look at Emerson's, this was a 2010 PNAS paper. This was the first paper which used genomic information to build a phylogenetic tree to delineate between mosquito species, between a mosquito complex where between cryptic species, and they did get high resolution. And this was one of the first demographic studies, evolutionary history studies, again done with genomic information, where they used where they tracked the evolutionary history of cattle. And now cattle today, 2 species of cattle which are commercially important actually evolved from their ancestral sequences. So basically, when you have genomic information, you have this opportunity to ask really complex questions. And I'll skip this for now. And so what we wanted to do, so when I first got into this research area, what I wanted to do is, well, okay, so if we have all of this genomic information, how can I use it to answer questions in biodiversity and conservation? And the first question that came to my mind was, because I was heavily invested in it, was why are there so many species along Anatolian mountains? So when we look at Anatolia, we all know that Anatolia is rich. It's a diverse, it's a diverse geographic region. But especially when you go along the mountaintops, we have this very high amount of diversity, not insects, with nearly all of these taxa. And what I was interested was in asking the question of these longhorn crickets. So longhorn crickets, because they're very easy to catch, they're very easy to sample, and they're also very diverse. So when you actually ask most biologists working today, they will tell you that the question of why Anatolia has a lot of diversity along its mountaintops is because of this one process called reproductive character displacement. And this process is very simple. So when you have, for example, 2 very similar species living in different geographic locations, as you can see morphologically, they don't have to be very distinct. They can be very similar to each other. So this is an example from famous Darwin's finches. As you can see, when you have similar species in different islands, they actually have similar bill depths, so similar beak morphologies. But when you put them in the same island, all of a sudden, as you can see, these 2 species have diverged incredibly. And you can also see a few hybrids between these 2. And so what's going on here is that if you have very similar species together on the same island, since they'll be producing hybrids that might be not, unfit hybrids that might not be able to survive, what evolution will do is selection would act directly on reproductive characters to quickly finalize speciation. So usually speciation occurs as a secondary event. So there's divergence for local adaptation and whatnot, and as a side factor, species diverge to such a degree that they cannot mate anymore. But in reproductive character displacement, selection directly acts on these traits that create reproductive isolation, so speciation is sped up. And this process of reproductive character displacement is usually given as the reason for why we have such high numbers of diversity in Anatolian mountaintops. And this comes from the effect, and Where does this theory come from? It comes from the glacial cycle. So as we all know, we have the world is in a glacial age, and we cycle through warm periods and cold periods. So when you look at organisms like these crickets, which are adapted to high altitudes, at warm weathers, they only actually survive at these, mountaintops isolated from each other. And they cannot really, they're basically up on sky islands. But when the weather cools down and these subalpine habitats, are able to lower themselves towards, lower elevations, all of these populations can mix together. And then when the warm weather hits, they're again isolated on mountaintops. And when the cold cycle comes back, they can again form larger populations at lower altitudes. So what you have is that you have these periods of isolation, with periods interrupted with periods of admixture, and then you have isolation, and then you have admixture again. And this is hypothesized to actually produce these patterns of reproductive character displacement. When this goes on long enough, all of a sudden you have populations that are, partially diverged, and when they come together, or species that are partially diverged, when they come together, they produce these unfit hybrids. So selection quickly acts upon this variation to finalize speciation. And there was actually 2 reviews on the subject, one by, Rashid, for example, where he actually talked about this process and also talked about these several hybrid tension zones in Anatolia where this process could be going on. So, but what I wanted to do was, this is a neat story, and we have a lot of patterns matching this story. So when you look at patterns of genetic diversity, we can actually see patterns that match this process of speciation. So, but what I wanted to ask was, using genomics, can we actually test some of the predictions of this process? And so before I get to this slide, let me talk about some of the predictions. So actually, reproductive character displacement makes 2 very strong predictions. The first prediction it makes is that divergence in reproductive traits should be much faster in relation to other traits. So traits that have not to do with reproduction, but to do with local adaptation should be diverging much more slowly than reproductive traits. So you should have an imbalance between reproductive traits and the other traits. So that's one real prediction of this process. And the second prediction is that divergence has to have a strong selective component. So it should not be neutral, and divergence should have a very strong adaptive signature. So we should have high selection coefficients for these kinds of divergence in these kinds of traits. So those were the 2 predictions. And the aim was, can we really test these predictions using genomic information instead of just looking at patterns of genetic diversity? So what we used was a system that I was, pretty knowledgeable about because I did my PhD on this, was a cricket species from the Kachkan Mountains. And when you look at these, they show the exact patterns that actually match that of reproductive character displacement. You can see we have 3 species, and one species is isolated by a geographic barrier, and then you have 2 species which are which show range overlap. And when you look at the species that show range overlap, they're genetically very similar, at least more similar, but morphologically they're very different, and sound is very different as well. And sound is very important in crickets because this is how they find their mate. So if there's a divergence in calling songs, there's usually speciation going on. As you can see, the populations or the species that show range overlap also have very different song patterns. But when you look at this blue species, it is morphologically very similar to the red species, and also it has very similar song structure. So you have this neat pattern where you actually do see what you expect from reproductive character displacement. And when you also look at morphological divergence, you can see that reproductive traits are really diverged in sympatry, and they're more similar in allopatry. But when you look at other neutral traits, they're actually more similar in sympatry and different in allopatry. So we have this very neat pattern that actually matches this process. But we wanted to ask, well, okay, the patterns match, but is the process really going on? And one way to test this is, so normally what you would want to do is you would want to set up an experiment and do a selection experiment. But when you're working with natural populations, it's not really easy to do that. For example, these kinds of experiments are really easy with model organisms, but when you have a cricket species that is, adapted to cold environments, you can't really take them in the lab and breed them and do experiments. So one way of doing experiments in the field is what we call the phylocomparative method. So this method actually uses phylogenetic trees to test processes about evolution. What it actually does is, so when you have a phylogenetic tree, and when you have trait divergence, you can say that if everything is neutral, trait divergence between species should match branch lengths of the tree. Basically, they should have because if we can take these branch lengths as proxies for time, if two species are separated, for a longer time, they should have diverged more. If they are, haven't been diverged for a longer time, they should be more similar. So basically, when you have an evolution tree, you can use this as a timestamp to test divergence between traits. So if everything is neutral, trait divergence should actually be proportional to branch lengths. But then you can actually fit different models. So this would be just a basic Brownian motion model. So you would only have two factors, your starting point. So there would be, value for the trait here. And as time goes on, this trait would diverge according to the amount of drift. So according to how fast mutations accumulate or drift accumulates on this species. So that's just a single model with two single values to estimate. But then you can also say other kinds of things. For example, if trait divergence doesn't actually match or is not proportional to the tree, then you can put in other factors like selection coefficients or optimum values. So you can say, for example, in this portion of this tree, maybe there's selection, so there's higher divergence than expected by Brownian motion. And you can try to explain that divergence by putting in a selection coefficient or a kind of optimal value for selection to go upon. So this is kind of a neat way to actually track evolution in the field without actually doing selection experiments. But the real problem with this is that you need a tree that actually models population divergence. And this is where genomic comes in. So if you have a tree or a single gene tree, and if that gene tree doesn't match your population tree, all of the inferences that you make from here will be wrong. So it's imperative that you actually model population divergence, because what you will be using are those branch lengths and that topology of the tree. So if your tree doesn't accurately represent your population, then all of your inferences will be wrong. And the best part about genomics is then today we can use information from hundreds of thousands of loci to actually get a great estimate of the population divergence. So basically here genomics comes into account, where we use all of that genome information to come up with the best possible population tree, so we can use to model divergence, speciation, adaptation, and selection. And so we actually did this. We sampled around 90,000 rad loci, and then we randomly selected 100 loci from this and built trees. So we basically used around 200, 300 loci to build different trees. And the best part of, again, using genomics is that you can actually repeat your analysis. For example, here's a population tree inference done from using 100 loci. Here's another one using another set of 100 loci. And as you can see, they actually converge on the same tree. So this is a good way of testing whether you've estimated a population tree accurately. Because if random gene samples give you the same result, then it's a good indication that you actually have accurately estimated the population tree. And so basically what I'm trying to tell you here is that when you have genomic information, you can actually just sample the genome different times and try to come up with good estimates of the population of your species tree. And if different gene sets actually converge on the same results, then you've actually validated your population tree. So this is actually a good way of doing repeated analysis. And so basically what we did was that once we were confident on our population tree, then we started testing some of these factors that we were looking for. For example, when we compared traits of evolution upon these evolutionary trees, we saw that the reproductive traits were actually diverging much more faster than other traits which were not under selective pressures, at least presumed by this process. So our first prediction from the reproductive character displacement was actually very clear from our inferences that actually reproductive traits were actually evolving much faster than other traits which were not under reproductive pressures. And then when we actually compared models. So for example, what you can do is you can compare different divergence models. Here, this is the drift model, so just a neutral model of divergence. Here we have a model of divergence with one adaptive peak. So there's selection, but selection is the same everywhere. And here we have a model with two adaptive peaks, which means that there's different selection for species which are in range overlap and different selective pressures on species which are in allopatry. And when we look at trait divergence for non-reproductive traits, we actually see that they cannot be differentiated from neutral diversity. So it just means that traits that have not to do with reproduction are actually evolving in a neutral kind of way. So natural drift is able to explain that divergence. But when we look at reproductive traits, we can see that the double adaptive peak model is a much more better fit, an explanation for the divergence going on. So this is and we can actually go ahead and measure selective coefficients. What this also shows us that to be able to see this amount of divergence in these reproductive traits, we need both high selection and high drift. And this is also fits in very well with the model of glacial expansions, because as we know, glacial cycles produce moments when there's high drift, so when their species are isolated on mountaintops, and then there's periods of high selection when they come together in admixture. So when we look at what this inference is telling us is exactly what we made from our predictions. This tells us that there is selection going on, and also tells us that to reach the amount of divergence that we see in this amount of time, we need both periods of high drift and periods of high selection, and we can actually quantify these processes. So basically, using this genomic information, we were for the first time able to actually show that selection and predictions expected from this process of speciation had actually occurred in Anatolian mountaintops. Now, with saying this doesn't really mean that this is a common process. So this is just one example, and to actually really solidify the conclusion that this is the main reason why we see a lot of diversity in Anatolia, especially in Anatolian mountains, we need examples from other species, other populations, other taxa to actually see if we actually, see the same results. And if anyone wants to ask questions, of course, they can always jump in, so no problem there. Okay, so that was biodiversity. So that's what you can actually use and I tried to concentrate more on natural species. For example, that's a very good way of anybody can actually do that type of analysis. Go to the field, sample populations, get genomic information, and do phylocomparative methods to actually measure processes of speciation and processes of diversification. So now on to another topic, which is conservation, and which is the one which I'm actively researching. So how can we use this information for conservation biology? And so in this instance, I will give an example from something I did in the United States, where we were actually tasked with helping to save an icon of conservation biology, and which is the devil-souled pupfish. Now, I don't know if everybody knows about this fish, because I certainly didn't. So this is, this is the Mojave Desert. So this is one of the hottest regions on Earth. And it's also called the Death Valley. So basically, it's very hot and it's very hard. And this Death Valley is one of the largest deserts in the world. And in this desert, there's a large oasis right around Las Vegas, or right around here on the corner of this huge desert. There's this one single oasis, and it's always been a very important, because this is where most of the diversity in this desert survives. So in this oasis, there's this single cave, and in this cave there's this one single population of fish. So this is a species called devil-souled pupfish. Its Latin name, I think, Cypredon diabolus. So its Latin name would be Cypredon diabolus. And this fish has just one population. So this species only has one population in this hole. And there's about 100 to 50 individuals. And the real interesting thing about this is this area is about 2 to 5 meters. So in this 2 to 5 meters pool in the middle of the desert lives this single population of fish. And there's only about 100 of them. And the remarkable thing is, they have been able to survive in this hole for over 20,000 years. So they have been isolated from every other population, every other species on the planet. They've been isolated from every other, relatives they have in these other marshes in this one single hole for 20,000 years. So that is why it's been called the rarest species on the planet. It's also sometimes called the toughest species on the planet because temperatures in this water can reach 40 degrees and it's very salty and there's usually a lot of seismic shifts. So this is a very active tectonic field. So there's really mini tsunamis for this fish. And they only have this one small area, which I'll draw here, this really small area where they can use to actually, sorry about this. Stop. Okay. They have this very small area where they can use to lay eggs and spawn and feed. So, the pool is, this area of the pool is just for swimming. And this is the only area where they can actually feed and lay eggs because this is the only area which sunlight actually reaches. So this is like very harsh conditions. And somehow this single species, isolated from everything else, has been able to survive there for 20,000 years. So it's just this remarkable story, this remarkable evolutionary story of how this fish actually survived for so long in this very limited environment. But that's not it. There's also something else going on because this population is also a cultural heritage. So when when we go to the map, you can actually see where the ash meadows is. So this is where the fish is. And this is actually, and right next to it, somewhere here on the map, is Las Vegas. So, we all know what Las Vegas is. And Las Vegas is this huge, city built in the middle of the desert. And also it's a place of tourism, it's a place of gambling and all that, and it's a place of entertainment. So there's population growth. And when there's population growth in the middle of the desert, there's demand for agriculture, demand for food, and demand for water. So what happens is, of course, when you're building Las Vegas nearby and you have this huge oasis with this huge amount of water right under it, so because all of this oasis is actually an aquifer, the really simplest thing is to pump water from ash meadows or from this oasis into the farms and agricultural communities of Las Vegas. And so basically that's what they started doing in the 1970s. But when they did that, automatically the water levels in the hole started to drop and this population numbers dropped from 100 to 30. So they were dying out. And this really led to a huge uprising from the environmental and conservation community where the legality started. And this was the first real environmental act that started perhaps the whole initiative. And people really took sides. I don't know if you can read, but there were people who had bumper stickers saying save the pupfish. There were other bumper stickers saying kill the pupfish. It was all over the news. And the local courts could not really come to a decision whether pumping of this water should stop to actually preserve these 100 fish, because most of what the community was saying is, we don't really care about 100 fish stuck in a hole. We need to feed this populace. Others were saying that this is a very important evolutionary species that we have to conserve. And this weighed its way up to the Supreme Court. So this dispute wasn't settled easily. It lasted about 4 years and it went all the way to the Supreme Court. So this was the first environmental act that made itself to the Supreme Court. And in 1978, I think, or 1977, the Supreme Court came to this landmark decision that it said that in 1952, we enacted the Conservation Act, and any species under the Conservation Act must be protected. So even if you want to pump water from this aquifer, you can only do it in a way of not endangering the fish. So this was perhaps the first, major victory of the environmental movement, and it has served as a rallying point for a lot of environmental initiatives. So not only is devil's hole pupfish a very important species from an evolutionary concept because it survived in this very harsh environment for 20,000 years with really no inflow of the genetic diversity, but it's also this very important cultural icon. But when you're a cultural icon and when you're famous, people start doing studies on you. So the basic real question was how did this fish get into the hole? Well, what was pursued was, we can see that this place is a desert today, but in the past, about 20,000 years ago, in glacial times, this place was filled with lakes. So this was actually filled with, so what we see as a desert today was actually filled with water. So what happened was when, when global warming came into effect, so when the climate started to warm, water levels dropped, and when water levels dropped, this fish somehow became isolated in this pool. So 20,000 years ago. But when people started, entertaining this, when they started looking at some genetic information, what they saw was that this fish wasn't really 20,000 years old. And there was, first genetic information from 2 studies actually indicated that this species was only about 200 to 100 years old. So basically, the fish hadn't been in there for about 20,000 years. It had been in there for only about 100 to 200 years. This also meant that this fish couldn't have naturally gotten into there. So it wasn't actually a relic from the past, but most probably someone had put the fish in there. So the real working hypothesis was that we knew that Native American Indians in the area harvested these fish for food sources. So someone had somehow put that fish in that small hole for some reason. And that was the reason why they were there. So they weren't an evolutionary anomaly. They hadn't survived there for 20,000 years. There was really nothing special about this fish. And it had only been there for 100 years. And it was actually, an accident, an artifact of human activity. Well, this caused a lot of problems because the federal government spends approximately $4.5 million each year to protect this fish. So there's a lot of funding, so a lot of tax money going into this. And if you know anything about the United States, everything is managed by taxes. So if you have, an important species that you're conserving, you're paying taxes for that. So this really was quite a problem for the federal government. All of a sudden, this species, which was not really important, which was not, evolutionary important, was just like this human accident. Someone had put it in a pond 100 years ago, and they were spending $4.5 million protecting it. So this was a huge problem. So what happened was that we were contacted by the Fish and Wildlife Service, so which is kind of like, the administrative branch which overlooks all conservation in the United States. They told us that we're in trouble. We have the species, we're spending $4.5 million, and just, we have all of these publications coming out telling us that this species is not important. So what can we do? Can you help us? And we said, like, yeah, we can't really help you, but if you give us, these fish, and if you give us funding to do some, extract genomic information, we can maybe ask the question of when this fish actually got into this hole. So whether the conclusion of these first initial studies was actually accurate. And again, the real important thing is when you have genomic information, you can actually model a lot of the different things. So the first thing we saw when we did our genomic analysis was that these species were really different from each other. So this is the Ash Meadows pupfish, its most recent relative, and this is the devil-souled pupfish. So we know that devil-souled pupfish diverged from this species, green species here. And if these species were only 200 years apart, we should really see them much closer. There's no way that they're different. But the real great thing about having genomic information is that it allows you to test hypotheses. And the hypothesis that we wanted to test was, we know nothing of the species, but we know something about vertebrate divergence rates. So we know how the genomes of vertebrates actually evolve. And so we said, using our genomic information, can we test different, models of evolution? And what we wanted to predict was, for example, given that this pupfish had diverged 400 years ago, what would be the genome-wide mutation rate? So we know something about the genome-wide mutation rate of vertebrates. So we actually wanted to test that. We wanted to test, given that this species diverged like 200, 400 years ago, what would be the genome-wide mutation rate? Given that it was diverged, let's say 20,000 years ago, what will be the divergence rate? And all of those kinds of questions. So we built 4 different models of divergence to actually predict genome-wide mutation rates. So here's our models. So this is where the pupfish diverged 60,000 years ago. This is where it diverged around 30,000 years ago. Here again, where it diverged, here it's around 6,000 years ago. Here it diverged around 20,000 years ago, and here it diverged around 400 years ago. So the question was, what is the genome-wide mutation rate when we have these, possible divergent histories? And when we look at our conclusions, we can see that if this fish had only diverged 200 years ago, its genome-wide mutation rate had to be on the order of 10 to the negative 5 per year. So that's a mutation rate on the order of viruses. So basically what our model tells us, if this fish had diverged only 200 years ago, the genome-wide mutation rate had to have matched the ones of viruses. Now, this is certainly not possible for any vertebrate species for it to evolve that quickly. When we looked at other models which said between 6,000 and 20,000 years ago, we had mutation rates on the order of 10 to the negative 7. Now, this is not unheard of, but this is very fast. So the only matching mutation rate for vertebrates we see that are this fast is actually in cichlid fish, which we know have super fast divergence time. And the other model which we looked, which was said at 60,000 years of divergence, gave us a mutation rate of 10 to the negative 8, which is the average mutation rate of vertebrates. So basically what we had was these two models in effect, which said we had to choose these. These were fast mutation rates, but it could be possible for vertebrates. This was actually the best prediction based on average mutation rates. But what more can we say about this? Again, if you have genomic information, then you can start looking at the genome for other different signatures. And when you have information from a lot of genes, what you can do is you can actually look at outlier loci. So we did this. So what we did was we scanned all of the genome looking for these really outlier loci. So as you can see, most loci are very similar in divergence patterns to each other, but once in a while you have these loci which are really, really different. And when we scanned the genome, we found about about 70 to 100 of these loci that were really different from all the other rest. And when we looked at these, all of these gave the same divergence pattern. So this is what we see from whole genome information. We know that the pupfish is the most recent ancestor of the green species, our pupfish, and the blue species is more distantly related. But when we looked at these outlier loci, what we saw that was some of these green individuals were actually more closely related to the blue species. So what this suggested was somewhere in the past, perhaps there was migration between the blue species and the green species. And we wanted to test these patterns. And then we tested this pattern using a migration, using the Abababa test, which is actually a migration test using whole genome information. And what the Abababa test does is very simple. It tracks novel mutations. So for example, if there's a mutation that occurred here in the past, before these two species split, this mutation could occur here, it could occur in this branch, it could occur in this branch. But if you have a mutation that occurred here, for example, in this branch, the only way that you see this mutation in these A and B populations is if there's migration. So if there's ancestral polymorphism, you can see that mutation anywhere. You can see that pattern of diversity in any of these. But if there's a novel mutation here, the only way that this novel mutation that occurred in this branch could spread to these populations after they've been diverged is by migration. So that's what we did using genomic information. We tracked the novel mutations on this branch to see if we could see these novel mutations either in the green species or in the red species, which is our. And what we actually found is that all of these novel mutations always occurred on the ash meadows pupfish. And when we tracked this, the timing of this migration, it was 20,000 years. So what this analysis tells us that 20,000 years ago, there was migration from this blue species here to the red species, but we could not see these migration patterns in our devil's hole pupfish, what this told us was that if 20,000 years ago divergence had not happened, so if this fish had not been isolated 20,000 years ago, we would have seen the signature of migration. Oh, okay, sorry about that. So okay, I'll repeat it again. So this is the Abababa test. And what this test actually does is it tracks, so on a given evolutionary relationship, it tracks novel mutations along the branches of a tree. So what it does is, for example, if the mutation occurred here in the ancestral population, then this mutation could end up in this branch, it could end up in this branch, or it could end up in this branch. So this mutation could actually end up in any of these populations because it occurred way in the past before these populations diverged. But if a mutation occurred on this branch here, a novel mutation occurs here, Since these species have already diverged, the only way that we can see this mutation in this branch or in this branch is if there's migration between populations. So basically, this is what the Abba-Baba test does. It tracks mutations, it tracks novel mutations occurring in this branch to see if we can observe, these mutations in this branch or in this branch. And when we looked at our results, we saw that all of the novel mutations here occurring in this, in this species, so in this blue species, we could find some of them in this green species here. But we could never find any of them, in devil's hole pupfish. So none of the mutations occurred in devil's hole pupfish. And this indicated to us that there was some sort of migration from, the blue species here to the green species here. And when we track the timing of this migration, really? Okay, I'm trying to draw it. Sorry about that. Okay. All right, so let me backtrack this way. Oh, okay. So someone are getting it. Okay, interesting. Okay, I'll just repeat. So I'll just repeat very shortly. I can't go over the whole thing. But what I can say is when we did a genomic test of migration, we saw that there was a migration from the blue species, this guy, to the green species here. So we observed migration from the blue species to the green species. But we could observe no migration from the blue species to the red species, so no migration. So there was no migration to the red species, to our species in the whole, but there was migration from the blue species to the green species. And we tracked this migration down to around 20,000 years ago. So 20,000 years ago, there was migration from the blue species to the green species. What this told us was that if this population had not diverged 20,000 years ago, so if Devil's Hole was a very recent divergence, then we should have seen this pattern of migration in Devil's Hole as well. What this actually told us was that Devil's Hole pupfish had been isolated pre-20,000 years ago. So what our mutation modeling approach and also what our migration modeling approach showed us is that Devil's Hole pupfish had been isolated in that hole over 20,000 years ago. So we could give a definite argument that that fish had been in that hole for at least 20,000 years, actually perhaps even much more longer. So if you look at genome-wide mutation rates, what that actually tells us is that fish has been in that hole for around 60,000 years. So that's really very different. So when we published these results, of course, the federal government was very happy because it made the fish species even more iconic, which is kind of a double-edged sword for me because I never thought I would make the federal government of the United States happy. But this is again a very nice example of how you can actually use genomic information to actually infer, at least help conservation or impact conservation policies, or actually, inform conservation policies about what they should do. Because if you have genome information, you can track things like mutation rates, genome-wide mutation rates. You can look at outlier loci. So you have a lot of this information that you can use to answer different questions. And the last example I want to give before wrapping up is perhaps the question that most people are really interested in, is that when you have genomic information, you can actually infer something about adaptations or adaptation processes. And this can be really important for informing conservation decisions. And this is something that's really looked over a lot of the time, because what evolutionary biology tells us, it can tell us about the process of adaptation. And that can be very important for basing conservation decisions on. And this is a field that is not really emphasized enough. So there's a lot of conservation studies going on using genetics, but very few of them use information from adaptation to inform conservation. So the question here is about these Pacific salmon. So this is another study we did along the North American Pacific coast. We all know what salmon are. So salmon are this very, very famous species because they're very charismatic. So what salmon actually do is we know that they're born in fast-flowing rivers, usually high up, up on mountains, and when when they mature, they migrate to the ocean. So they start off in rivers. So all of these salmon start off in rivers, and then they migrate to the ocean, stay for 1 or 2 years in the ocean, grow really large, and then they can perfectly track, from the ocean. They have this homing ability to actually migrate to the exact same location which they spawned in. So that's this remarkable species. And what they can actually do is they can actually jump up off rivers. They can just like go opposite to flowing rivers and jump off in these like really lovely avalanches and go to those rivers which were they born in. So these are really famous and charismatic species. Yes, yes, Adam. Yeah, it's related to a lot of things. Well, yes, it's actually has to do with resources. So what it is usually salmon have high population numbers, and these rivers are not really that big. So when you have a lot of populations trying to, grow to sizable maturity or actually have enough nutrients to mate, it's really problematic. So what they do is they migrate to the ocean and they use a lot of the resources in the ocean to actually reach larger sizes. And then what they actually do is they bring some of those resources back to the rivers which they're in. So it's like this really nice symbiotic relationship where some of the resources in the oceans they actually bring back to the rivers as well. So it's this very neat kind of system, but it's basically to grow. So the ocean is much more larger and they can grow to much larger sizes in the ocean because there's less competition and more resource. So that's the main reason. As for the homing ability, I don't really know about the physiology of how they home back. I think it has to do with smell because salmon can really smell. I think the smell sensors of salmon are incredible. So I think the basic cue for them actually finding exactly where they spawned is using olfactory cues, but don't quote me on that. I'm not really sure of that. But as I said, it's this very important species, and it's also commercially very important because it's a very good food source for us. But it's also this very nice, story where these salmon actually migrate back in this really perilous adventure to go back home to where they spawned. And the other fascinating thing is, of course, they only mate once. So what salmon actually do is they climb off of these rivers back to their homelands, and they mate once and then die. So it's yeah, photosensitive could be. As I said, it's an area that I haven't really researched. But for our priority, it's just important to know that this is like a very important species, both from this nice evolutionary question and also because it's a very major food source. But the real problem is that there's a very distinct pattern of migration. So basically, usually, as we say, fish, they migrate mature. So they reach maturity and they migrate. And this migration usually happens somewhere around somewhere around between May and August. So between May and August, these fish actually mature, they go back to the ocean, and then they come back to mate again in the summer months. So what the pattern is, they mature and they migrate, and then usually during July, August, 2 years or 1 year later in July or August, they swim back up to their native habitats. Now the real problem is global warming. So when we have global warming, so when climate is actually heating up, what we have is we have reduced water flow during July and August. Sometimes because snow melt is much more, there's less snow, and snow melt is actually, occurs much more faster. So what you have is you have lower flowing waters. And when you have slow flowing waters and, waters with lower temperatures, then salmon cannot actually swim back to their native habitats. So because of global warming, all of a sudden you're having these population decline in salmon. They're not able to go back to their home, to their spawning sites as much as they could in the past. And so one really nice adaptation that salmon have built for this is what we call premature migration. So what happens is, and this is one of the best examples of adaptation to global warming, is some mature, migrate before maturation. So usually these species wait till August, July to mature and then migrate to the ocean. But what they do here is premature migrants is they don't wait, until they're mature. They migrate to the ocean before maturity, stay in the ocean, and mature and come back to the rivers during the months of May, somewhere around May. So what's basically happening is that they go to the ocean before maturation, but they come back early. And when they come back early, then they can use those waters. Waters are still cooler, they're still high flowing to come back to their native habitat. But then the problem here is then once they're back into their native habitats, they're not mature. So they have to mature in still water. They have to mature without the resources of the ocean. And so this is a very nice adaptation to actually, and they can do that. They have a certain adaptation which enables them to mature fastly in river waters. And this is a very nice example of an adaptation to global warming. So basically what we have in salmon today is we have premature migrants and we have mature migrants. And these premature migrants are very important because for the continuation of the species, it is a way of this species of tackling global warming. So basically, the important question from a conservation standpoint is, how different are these premature migrants to mature migrants? Are they different enough genetically so that we can actually conserve them differently? So this is when you look at genetic diversity. So we're back at looking at genetic diversity patterns. So we have different populations of salmon, and the triangle is premature migration, and the circle is mature migration. And as you can see, different populations have both mature and premature, lifestyles in them. And when we look at genetic diversity is not really, affected by whether the, individual is a premature migrant or whether the individual is a mature migrant. And actually, you can actually see that patterns have to do with geography. So red, green, orange, red, and blue and purple here. So as you can see, when you look at general patterns of genetic diversity, mature and premature migrants are not really different. And what really differentiates these populations is actually geographic structure. So conservation initiatives will build on this premise. It said that, well, okay, we can see premature and mature migrants in different populations. So this means that this adaptation is very easy. It occurs in multiple places. So we don't actually have to differentially protect these migrants. If we just protect, salmon in general, even if premature migrants, are eradicated, or if premature migrants go extinct, they can readily evolve again because, as we can see, they've evolved in a lot of different places. So this, life history evolution, or this life history trait, has evolved in multiple different populations. But again, this is just looking at genetic patterns of diversity, and we wanted to ask the question: well, is this true? What can we look at genomics? Can we find the gene regions that are responsible for this divergence, for this life history trait? And can we say something about the evolutionary history of that adaptation? So how that adaptation actually occurred. And when we looked, so what we did was basically we did genome scans for adaptation. And when we looked at the whole genome, most of the genome is very similar between the two life history variants. But in this one gene region, which is the GRABL1 region here, we have high amounts of differentiation. So this is genome-wide rates of diversity in premature and mature migrants. And as you can see, they're very similar. But in this GRABL1 region, in this gene region here, as you can see, there's a huge difference between mature and premature. And as you can see, genetic diversity in premature has decreased substantially. So this is a very good indication of a selective sweep, so that there's some sort of selection going on in this gene region. And there's a causative mutation in this GRABL1, which actually switches the population from a mature migrant to a premature migrant. And this GRABL1 gene is very interesting because we know from human studies that this has to do with development. So the GRABL1 gene region, for example, mutations in GRABL1 in humans, in Latin populations causes child obesity. So basically, when you have a mutation in this region, you accumulate fat much more quickly. And this really makes sense because premature migrants come to the rivers, which have less resources, without maturing, and then with limited resources, they can reach these larger sizes. And it's because of this mutation in the Graboban region, which enables them to accumulate fat much more quickly with less resources. This is also a very nice, fun evolution fact, which shows that the same mutation or the same kind of evolutionary process can be really detrimental to one species, like humans. In humans, it causes child obesity, so it's something that's not wanted. But in fish, it's actually a very positive mutation, which enables these fish species to survive global warming. So this just shows how mutations are really neutral. It's a very nice example of how mutations should be beneficial or detrimental. Mutations has totally to do with the context which they're evaluated in. So basically what we really saw was that there was this one single mutation in the Graboban region that switched the species from, the population from mature migrants to premature migrants. But when we looked at the evolutionary history of this mutation or of this divergence, unlike the others, we saw that if we build a tree off of this gene region here, as you can see this tree, I can't really draw because I'm out of the lines. These mature migrants from all of the other populations are branched in one ancestry, and premature migrants are, actually branched in one clade. So there's two different clades. So which actually this shows us is that this mutation or this region didn't really mutate, several times in different populations. Because if that was the case, you would see, premature migrants from the same area clustering again. But what we see is this really huge difference between mature and premature for this gene region. This actually shows that this was only a single evolutionary event. So this event happened once, and all of the premature migrants or that premature allelic variation is from these, from that very ancestral population. So basically, this shows us that this mutation didn't occur several times in different populations. It occurred only once and spread. So basically, this tells us that if we lose the premature migrants, there's no guarantee that we're getting this mutation back. So this makes a huge amount of difference. All of a sudden, just protecting any populations, it's not enough. We have to differentially protect the premature migrant. We have to protect this very important genetic diversity or this very important evolutionary diversity or evolutionary adaptation that came into part. So it's not enough just protecting the species. We have to protect this adaptive unit. And this is very important because going forward, it is very important for conservation initiatives to find these, adaptive variants that are able to buffer climate or any kind of change. So, changing the focus of conservation biology from looking at just grand patterns of diversity to actually finding these very important adaptive variants and concentrating efforts on these adaptive variants, which can actually, weather the storm, so to speak. So that's about it for now. Before, I hope I've been able to show you some examples where we can use genomics to answer questions of biodiversity, not just patterns, but processes, and also how we can use, genomic information to actually tell us something about or inform conservation policies, whether this be just by evaluating evolutionary history or by looking at the adaptive basis of populations. One quick question, so just wrap up. Again, as I said, evolutionary genomics is very important as an applied field because when we look at our society today, when we look at global problems, evolution is really in the center of all of it. So here's a nice example. For example, chronic disease. So today we're in problem with chronic diseases. We have a lot of chronic diseases. For example, we have heart failures, heart diseases. And one major reason because of heart disease is because we eat food with a lot of fats. And we can't really digest that fat because and the problem is we eat a lot of fatty foods today, but in the past we didn't have access to fatty foods. So we evolved to actually obtain fat from numerous sources, from plants, from other kind of seeds and whatnot. So all of a sudden, when you switch to a fat, hugely to a high-fat diet, all of a sudden you have this problem. And this is an evolutionary problem because we have evolved under low fat. All of a sudden we have high fat, so our genes are, are not ready to actually have all of that influx of ready fat. So that's an evolutionary mismatch that has to do with our health. We know, we have biodiversity loss, so global warming, or environment is changing very quickly, and we're also speeding up that environmental change. So populations cannot actually adapt as quickly to this change as in the past. Again, that has to do with evolution. So we have problems, in our food sectors. We're trying to feed the populace, and we have crops, but but we have insects that feed upon those crops, and then we have pests. When you have large, for example, crop fields, then you have a large population of insects, and then you have a problem. Then you have pesticides, and you try to combat that with pesticides, but then there's resistance. So when you look at all of these global problems in food sector, in health sector, or in biodiversity, it really all comes down to evolution. So having applied evolutionary genomics is very important, and genomics really helps us in that regard. So it actually helps us to build a field of applied evolutionary genetics, or an applied field of evolutionary biology, where evolutionary biology is not just theoretical, but it can actually help us in solving real life problems. And okay, so the greatest thing is that if you want to solve these real world problems, it's all about, as I said, fitness landscapes. It's all about adaptations. For example, here we have a population which is maladapted. For example, let's say under global warming, and we wanted to get into this fitness peak. So what we have to do is we have to understand the processes which will be able to move this population from here to here so it can survive. But we can also have the opposite problem. We can have, for example, insect populations that are resistant to most of our pesticides, and that they have high fitness, and we want to reduce it to this level so that we can combat them. So again, this is an evolutionary problem. So what we have to do is we have to understand the process of adaptation so that we can actually modify them through time to actually solve some of our problems that we see today. And sometimes this is very easy. For example, in the Pacific salmon, this was very easy because we got lucky, because adaptation was just due to this one single region here. But what we know about complex traits is that usually adaptation is not about a single gene, it's about multiple genes. So in the Pacific salmon, we were very lucky. We looked at it and we found this one major gene which was effective. So it's easy to understand the process. But is it always true? No. For example, when we looked at another fish species, this is the Masu salmon, this is way up in the coast of Japan. And when we look at the evolutionary basis of migratory behavior in these salmon, as you can see, they also have size difference. When we did, a genomic scan, as you can see, but this phenotypic divergence, there was not just one region that was informative. There seemed to be a lot of regions, and there really didn't seem to be one single gene region that was associated with this, pattern of life history evolution or with this divergence. So how do we tell apart? How do we tease apart from this which genes are important for evolution? So when we have polygenic evolution, when we have evolution or adaptation caused by multiple loci, how do we tackle that? And that is, again, the best part of genomics, because today what we can do is we can fuse quantitative genetics with genomics to actually come up with complex trait analysis. So what we can actually do is, instead of just looking for these one single region that has diverged, we can model all of the divergence of the gene and try to understand which regions are actually contributing to heritability. So basically what we can do is we can scan the genome for regions, how much each region is contributing to divergence, how much each region is contributing to that differentiation. So this allows us to actually just understand which regions are responsible for traits. So we don't really need to home into that one single. If we're not lucky, we don't have that one region that's differentiated, we can actually now scan the whole genome. And the reason why we can do this is, in the past, to do this, you would need experimental designs. Because to track evolution, you would need pedigrees. You would need to know who came from who, who's the parent of who transferred their genes from their offsprings and whatnot. So you needed to measure heritability, you needed to measure ancestry and whatnot. And what genomics enables us to do is that even if we have no understanding about a pedigree, we can actually build a pedigree de novo. So we can use genomic information, and this is a pedigree built de novo, from random individuals collected random in the field, and we can track their ancestry using genomics. And once we have information about ancestry, then we can track, the evolution, or actually track how traits diverge through time, and we can track heritabilities, we can track genetic variances, we can track how much of the variation is due to genetics, how much is it due to phenotypes. The genomic information gives us this ability to not only find, not only get lucky, but we can actually do quantitative genetics in the field. We can do quantitative genetic experiments using complex trait analysis, and we can actually understand how adaptation or how polygenic adaptation occurs. For example, the first examples of these can be found in human genetics. For example, the first study done with complex trait analysis was to infer body size, and we actually right now know how height in humans is an adaptive process, and how many genes and how many polygenic genes actually go to affecting height differences in humans. So it opens up this huge new field where you can actually do really complex analysis in any kind of population you want using genomic information. So I think I'll just wrap it up there. A few thank yous. So these are all of my agencies which have funded these research. Some of the people in Hacettepe University that helped through my research. The team we have in University of Utah, and the team I had in University of Davis. And to all of you, thank you for listening. And if there's any questions, I'm happy to answer. Oh, okay. So active genes or pseudogenes are worth looking. It depends on what you want to do. For example, so now the best thing about a genomics is, again, you can actually the trouble with. So if I understand your question correctly, I'm not sure how pseudogenes can inform us about conservation biology or any other process, but it can actually tell us something about genome architecture. So if genome architecture is important, for example, with species with a lot of pseudogenes, that just means that there's somehow there's a if I'm not mistaken, there's a difference in loss for words here. So mitosis or meiosis, sorry. So basically, how genomes evolve. So if populations accumulate a lot of garbage genes or pseudogenes which have no information, that just means that the genome is not enough to actually call unnecessary portions of the genetic diversity. So a lot of regions which are not used anymore are still kept in the genome, and that can inform you something about genomic architecture, but I'm not sure how that can inform you about conservation. But one important thing is, okay, of course, today we know that even what we thought as junk genes or pseudogenes in the past actually do have some sort of function. So they might have regulatory processes. And that also comes into effect with, complex trait analysis. So when you have complex trait analysis, you can actually see, you can scan all of the genome, and you can look for regions of the genome that are affecting adaptive divergence. And so it helps you in finding regulatory regions. So example, if you've scanned the genome and all of a sudden you're seeing these pseudogenes which are actually affecting heritability, which actually are affecting the evolution of that trait, you know that there's something going on in those genes. For example, that's one type of inference you can do. About what? There are some quantum biological approaches about some kind of migration patterns in other animal species like birds. I don't know if it's related to your genomic approach. Well, yeah, most probably it is the same approach. So the Abba-Baba test is actually a very good test of migration, but it's specifically built for tracking low amounts of ancient migration. So the Abba test is actually a migration test using the whole genome, so genomic information. And its specialty is that it can actually track very small amounts of migration, that ancient migration. For example, its first use and what it was actually developed for was for tracking migration in human ancestry from Neanderthals to Homo sapiens, or maybe not Homo. Yeah. Was there Homo sapiens back then? Maybe Dilek would know, but basically it had to do with tracking migration between Neanderthals and humans. And that was an ancient migration and the signature was low. So that's why the Abba test was developed. So it's actually a very good test of migration, and it has been used in several species from birds to mammals. And its real importance is that it can actually track small amounts of ancient type migration. So that's why it's been developed. And I hope that answers your question. Okay, so do you also look at transcriptome evolution, especially for the adaptive studies? Yes, of course. So that's another goal. So I have not studied transcriptomes yet, but I do have a project which, hopefully will get funded. So it's I'm writing it up at right now. So this project, which I didn't actually, I have, no, that's a different one. So this one here, we're trying to track the evolution of color dimorphism in crickets. There we will be using transcriptome data, and of course it's vastly important. So one of the greatest things about the genomic revolution is transcriptome. So because more or not, some of these adaptive processes is going to be phenotypic plasticity. So it doesn't always have to be selection. It doesn't always have to be some sort of structural adaptation, but it can also be some sort of plasticity involved. And transcriptomes is like very important in tracking that kind of evolution. For example, in the master data set in the fish where we didn't really get a good signature of adaptation, perhaps it's phenotypic plasticity, perhaps it's difference in expression levels that are causing divergence. So yes, transcriptomes are a very important part moving forward, especially if you find a lot of signatures in regulatory sites. That again, and also it enables you to test things like epigenetics. So basically how the environment influences, trait development. So it's also important in that aspect. Okay. But what will you measure in transcriptome? It changes all the time. Yes, of course, that's the whole issue. In transcriptome, it's the cells matter. So, but I think today what you can do. So I haven't really done too much transcriptome analysis. So if someone knows, they can always chip in. But you can you can look at whole transcriptome, or you can look at transcriptome of specific cells or specific tissues. So basically, you would have to have some sort of knowledge of what you're testing. For example, in my crickets, there's coloration, so perhaps I would be testing pigmentation. So, tissues, or, pigmented tissues, I'm trying to look for that kind of signal. So it depends on what you're actually testing. And the tissue does matter as far as I know. So you would have to have a good idea of the tissues that you would be using to test transcriptomes. And that's also why it's easier to do genomics than transcriptomes, because you would actually have to have, good tissues. So you would either have to fix certain tissues in the field, or like quickly get them into the lab before the RNA degrades. So, you have to have a better design, a better idea of what you're testing. But perhaps today things have changed, and as far as I know, you can have whole, whole transcriptomes, but I'm not sure how you use them in those contexts. Ah, yes, exactly. Yeah, definitely. I mean, you could, yeah, a lot of people are looking into that, to building, regulatory networks. But again, that's, I'm not sure how much that is applied to natural populations. So in conservation biology, and what I wanted to concentrate most was on non-model organisms and like natural organisms. And building regulatory networks for Arabidopsis or for Drosophila and whatnot is kind of easier, because you have good genomes, you have annotated genomes, you know the regions, and it's much more easier. But when you have de novo, populations which you have no prior genetic information, that kind of regulatory network can be much harder to build. But of course, it's a really important part of the research. So what I wanted to say there was, it's not about neutral evolution. So what I wanted to say, so I don't want to be misunderstood. So neutral mutation, so there's no such thing as a neutral. Neutral mutations is neutral according to context. So a mutation can be neutral today, but it can be adaptive in the future. So what we say by neutral, it totally depends on selection, or at least the environment which is in. But what neutral actually means is that there's no adaptive process on it. So it just, the occurrence of that mutation just depends on, the random process of, DNA replication errors. So that's what a neutral mutation is. So it's a mutation, it occurs through natural, DNA replication error. And since it's not beneficial, or since it's not detrimental, nothing happens to it, it stays. But what I meant by, positive or detrimental was according to the context. So for example, in the Grabal1 region, the same region, mutations in the same region cause human obesity. So it's a very detrimental trait in humans. It causes child obesity, and if it's, and it can cause a lot of health problems and early death. But in salmon, it's an adaptation to global warming. So what I meant by that was the same mutation or the same variation, depending on one context, can be beneficial for the organism. Depending on another context, it can be detrimental. So that's what I meant. So there's like, mutations doesn't, so that's what I kind of wanted to make in that point, was not that, each mutation, whether it's beneficial, whether it's adaptive or maladaptive, depends on the context. Yeah, yeah, definitely what Andres says, yeah. How do you explain Darwinian selection to conservation? What do you mean by explain? Use Darwinian? In what context do you mean? I mean, how can we use Darwinian selection to answer conservation? Or I don't really understand the question. Eden, could you try to clarify the question? Does selection affect current speed? Yes, of course. I mean, that's the whole issue with, let me see. This slide is, that's what I wanted to actually come to. Yeah, it's all about this fitness landscape. Definitely selection affects current species, because, that's what I meant. Because, selection is going on all the time. So basically, when there's environmental change, or when you're in an environmental context, when you're a natural species, there's always going to be some sort of selective pressure. And why it's important for conservation is to understand where in this fitness landscape our population that we want to conserve is. For example, if the population that we're trying to conserve is here, when we look at it, it says, so this is, its fitness landscape, and it's way above its fitness landscape. That means that this population is in good order. So, there's most probably balance in selection to keep it at this peak, and, there's no problem, whatsoever. But for example, our species, instead of being here, our species could be here for a given environment. So as you can see, for the given environment, the optimal fitness is here, shown by this red circle, but our population is here. So this would mean that our population cannot really adapt to this fitness, and there is selection for it to go between this. And the question is, how can we speed this selection to conserve the species? Yes, selection will work if the environmental stays, and if this species has enough genetic variation to adapt, slowly by slowly, it will climb up this fitness landscape. But for conservation purposes, is there any kind of strategy we could actually, do to help this population reach this fitness, goal much more quickly? So, having this information, knowing how selection works on a genomic level, knowing what genes, what processes actually, what process is actually ruling this change helps us to actually modify or help this species get to this fitness. But selection goes on itself. The real important question about conservation is how understanding that selective processes, understanding genomic processes that goes on, so we can aid populations that we want to conserve, if we want to do such a thing. That's a debate in itself, whether we should actually butt our heads in. But that's a debate in itself. Yeah, exactly. So that's the whole issue, and that would be here. I mean, that's the whole thing with, for example, pest insects, as diseases and whatnot. What we want to do is we want to lower their fitness. Again, it all has to do with understanding genomics, understanding how species adapt and how, through time. So understanding this very intriguing connection between phenotype and genotype in environment. And genomics can really help bridge us that gap. Again, it's the same thing. So a disease would be here. So, for example, what we want to do with tuberculosis or AIDS and whatnot is we want to reduce its fitness. We want to get its population down to this, so it won't really affect us much. But again, we need knowledge about how it evolves, how it responds to environments, how it responds to different selective agents, so we can modify its journey in this fitness landscape. Exactly. But whether we should do that or not, again, it's a debate. Obviously we do it for human health reasons. We do it. We combat diseases. For crops, pesticide, resistance. Right now we can actually engineer, pest-resistant crops and all that. So we continually modify it.
Hi, me and my nosy.
Hi.
Thank you.
Are we finishing up? Okay.
Ismail.
Dilek, I'll just answer this question, then we can finish up. Do we
Okay.
dare to eradicate species? I mean, what about ecosystems? Yes, exactly. It's never a good idea to eradicate species. Another portion of this is, of course, ecosystems. So there's a whole new branch of that. So what you can do is, using genomics, you can do eco-evo. So in the past, this was really hard to do. So there's another thing called eco-evo, evo dynamics. So how ecology affects evolution, but also how evolution affects ecology. We're doing this all the time. For example, especially today, because of changes, there are certain species that can adapt to human environments. Like mammals can adapt to human environments very fast. Birds can adapt to human environments very fast, and this creates some sort of imbalance in the ecosystem as well. So this would be kind of feedback between evolution and ecology, and that's a whole different area. And again, genomes can help us in understanding, how quickly that feedback can happen. And today, from some preliminary studies, we know that evo-eco is also a very fast link. So if you have, rapid evolutionary divergence of certain species, this really affects the ecology or the ecosystem, and it can imbalance the ecosystem. We have, for example, this issue we have going on, in a research we're doing right now. For example, and I'll wrap this up, this bear species, for example, today, when we look at, so this is, Turkey's Eastern Turkey, when we look at the bear population in Eastern Turkey, we have a huge bear population, but we have nearly no prey species. And we actually have a very large carnivore population in Eastern Turkey without prey species. What's happening is that these animals, bears, wolves, and lynxes, they're getting adapted to human resources. Since we have large city dumps, since we have lots of food, extras, these animals, which are, pretty smart and cognitively complex animals, they can adapt very quickly to these new resources. So all of a sudden, you have this major imbalance. You have these species which are getting anthropomorphized. So they're becoming, anthropogenic animals. They're kind of like being domesticated very quickly because they're using more and more human resources. So their populations are growing. And all of a sudden you have this imbalance with very few prey species, a lot of large carnivores roaming around in the area. And that really affects ecosystems because these are top down. So you're talking about the highest, link in the food chain, which will affect ecosystems. So understanding how these animals actually adapt to these urban environments is very important. And this is another project we're doing with Can Şekercioğlu right now, is to look at how brown bears and wolves and lynxes adapt to these human resources and to see how fast this adaptation is going. For example, Can and his team discovered a population of sedentary bears. So we have bears that just live off of city dumps. They don't migrate. So normally bears, as you can see with the black ones, they migrate all the way up into Georgia and all the way, which is normal for bears. But then we have this gray population here, which doesn't even migrate at all, which just lives off of cities. And this population is growing. So exactly, it's very important to actually also understand the ecosystem implications, and genomics can help us in, usually it was always ecology, evolution. So ecology changes, evolution changes. Today we know that rapid evolution changes in single species can actually affect ecology and ecosystems, and that's another avenue of research. And with that, really, thank you a lot for listening, and I hope this has been a nice talk. See you guys soon. Thank you, guys. Dilek, you can finish it off, I guess.
Hi, thank you so much for this well presentation, and also thanks so much to accept our invitation to be webinar presenter.
Always.
And thank for all of you to attend our webinar session today. A recorded version of this webinar will be available. Yeah, we are looking forward to see you all in next webinar.
Okay, thank you all, you guys. Thanks. Bye-bye.
Bye.
Bye-bye.